Heredity and Evolution. Lecture number 1. So, as the name suggests, in this particular lecture, we will be learning about the concepts related to heredity. Okay? So, let's go on with the introductory part. Fine. We have seen that reproductive processes give rise to new individuals that are similar but subtly different. We have also discussed of how some amount of variation is produced even during sexual reproduction in the previous chapter. The number of successful variations are maximized by the process of sexual reproduction. Here we will be learning how these variations are getting induced in the child. So, accumulation of variation while reproducing. Look, the inheritance from the previous generation provides both a common basic body design and a subtle changes in it for the next generation, say generation number 2. Now, what happens when this new generation, that is the second generation, undergoes reproduction to give rise to a whole new generation? Let us name it as generation number 3. So it was actually found out that the new generation, that is the third generation, will have differences that they inherit from the first generation, that is generation number 1, as well as newly created differences. So these are the 3 generations, and shown below are the differences in colors. So depending on the nature of variation, different kind of individuals would have different kinds of advantages. So selection of variants by environmental factors forms the basis for evolutionary processes, as we will discuss later in the sections of this particular lecture. Okay? So now let's come to a whole new important concept known as heredity. Okay? Now we are entering the core part of this lesson. So let's start with heredity. So in a simple language, heredity is a biological inheritance of the characteristics from the parent generations. So for example, consider these 2 years of the children. In this case, these ears are somewhat not alike. You can clearly see that in one case the ears lobes are attached and in the other case they are detached. Please have a look carefully. So to get a much better clarity of why this happens, a scientist named Mendel has conducted experiments on flowers to understand the distribution of the parental characteristics among the generations. So the scientist was Johann Mendel. Okay. Now let's study about the experiments conducted by this person. The experiments conducted by him were named as the Mendel's experiments, and we will be learning about them shortly now. So Gregor Johann Mendel carried out several experiments on pea plants. He carried out large number of monohybrid and dihybrid crosses using many contrasting characteristics and put forward several important conclusions. Okay. I hope it's clear till now. So here you'll be seeing how 2 different pea plants were crossed with each other twice, and you can see the 3:1 ratio in the third generation. Okay, in the third line. Now let's go in detail. considering the same image on the top left of your screen, in this experiment, Mendel has crossed a plant with a purple flower and a plant with a green flower belonging to the parent generation. In the next generation, all the plants had purple flowers only. Okay, please observe this carefully. And when he crossed the two purple flowers of this generation, which is F1, he found that 3/4 of the plants were purple and 1/4 of the flowers were green. So it was something interesting here. Note the 3:1 ratio of the purple is to green flowers. Okay. So similar experiments were conducted with different type of plants. So he took a tall and a short. In the first generation he got all tall. And in the third generation he got 3 tall and 1 short. Fine. It was similar to what has been explained ago. So to have a better explanation of what is going here, what he actually did was, he crossed a tall plant and a short plant. In the first generation, he got all tall plants in the new F1 generation. Okay. So when he crossed the two tall plants of the F1 generation, he got a 3:1 ratio of the tall and short plants in the F2 generation. So the ratio of the type of the plants was 3:1. That is, the tallest to short ratio was 3:1 in the F2 generation. Now, what are this P, F1 and F2 generation? And what is a monohybrid and a dihybrid cross? So let's go in detail of this. So let me define these new generations for you. So coming to the definition section, the P generation is nothing but a parent generation. The F1 generation, it is a generation resulting immediately from the cross of the first parents, okay, first set of parents. Then the F2 generation, it is the offspring resulting from the cross of the F1 generation. So the basic cascade is as follows: P followed by F1 followed by F2. Okay. Now let's define a trait, trait. So it is a character which we see in a plant or a species. For example, tall, short, dark, fair, etc. Now let's define a genotype. I repeat, genotype. Genotype. So it is a representation of the type of the genes. Okay. For example, in the previous experiment, capital P was purple and small p was green. Here capital P and small p are the genotypes. In the second experiment, double capital T was tall and double short T was short. Similarly, double T and double small t are the genotypes. Now, let's define a phenotype. Okay, it is a representation of the physical expression of any organism. For example, in the previous experiments, purple, green, tall, short, etc. are all the physical representations of the plants, which are nothing but the phenotype only. Now, let's define the monohybrid species. So, those species which have only one gene in their genotype are called as a monohybrid species. For example, in the first Mendel's experiment, the purple flower had capital P and the green flower had small p. As they have only one gene, they are a monohybrid flower, a species or a plant. Okay, they are monohybrid. Similarly, we have a dihybrid, trihybrid, etc. Now we will be defining a dihybrid species. So those species which have two genes in the genotype are called as dihybrid. For example, in the second Mendel's experiment, the tall plant had a double T and the short has a double small t. As these genes have two, generics. These are called as a dihybrid species or a plant. Now let's come to the concept of dominant and recessive traits. So consider the Mendel's experiment which we previously discussed. So in the first experiment, we have represented the purple with a capital P, purple flower with a capital P and a green flower with a small p. So here the capital P represents that it is dominant and the small p represents it is recessive. Okay. So one important observation you are expected to make here is the capital letter represents the dominant trait and the small letter represents the recessive trait. I repeat, the capital genotype represents the dominant one and the smaller one represents the recessive one. Sometimes we may have a combination of both. In that case, the dominant one plays a major role. Now, let's come to genotypic and phenotypic ratio. So starting with the genotypic ratio, it is the ratio of all the different types of genes. For example, in the first Mendel experiment, the genotypic ratio in the F2 generation was 3:1 because we had 3 purple flowers and 1 green flower. So please have a look at the diagram. We had 3 purple and 1 green. So 3 capital P is to 1 small p, which is 3:1. Now similarly, let's define a phenotypic ratio. So phenotypic ratio is the ratio of all the different type of phenotypes. For example, consider I have crossed a round yellow seed and a green wrinkled seed, considering the round yellow to be dominant. So for a clearer idea, let's move on to the next slide. You have a huge diagram which will help you understand it better. So here I have a round yellow which is dominant given in capitals and a green wrinkle which is given in small letters which is recessive. So when I cross this both, in the F1 generation I get a round yellow seed with both the genes. Now when I cross 2 seeds of this F1 generation, I get many seeds in the F2 generation. Of this I have 9 seeds which are round and yellow. Okay. These are 9 in number. Similarly, I have 3 which are round and green. These 3 are round and green. Please note the genotype. Okay. Now I have 3 wrinkled yellow seeds. Okay. 3 wrinkled yellow ones. And I have 1 wrinkled green seed. Only 1. So in this case, my ratio is 9:3:3:1 for both the genotype and the phenotype. Okay. So the continuation of this lesson can be found in the next lecture. Thank you for watching.